Good morning. My name is Lucas Santos, and I'm from Brookline High School, and I'm here today with my lab partner, Dominique Dang, from North Quincy High School. And we're going to talk to you about our research focused on the biomechanical characterization of the porcine or pig tricuspid valve. Okay, now let's get into an introduction of what the tricuspid valve is and what it's composed of. So valves in general are important to a healthy heart because they prevent backflow and allow blood to only flow in one direction. So the tricuspid valve coordinates blood flow from the right atrium down to the right ventricle and up into the pulmonary trunk. The tricuspid valve has three leaflets, the anterior, posterior, and septal. And the anterior is normally the largest and the most uniform, so we focus on that in our studies. Now, the tricuspid valve is composed of long strands of molecules that are in its connective tissue matrix. Two very important components of the tricuspid valve include elastin and collagen. Elastin allows for the tissue to stretch and gives it that elasticity, and collagen allows for it to compress back to its normal structure and give it that framework. Radial and circumferential fiber alignment is not symmetrical throughout the tissue in the tricuspid valve. So we are interested in measuring the radial and circumferential stretch and strain with different loads.
So now just a little bit of background about why we started this experiment and our main question. So fresh tissue mechanical tests have lengthy preparation time, such as the ones doing at our lab that are ex vivo. So you can see in this video, that's the pumping heart. So preparation, for example, like this experiment take over 5 hours. So our main question is, do the mechanical properties significantly change during this time? So our goal is to characterize the mechanical properties of ex vivo porcine tricuspid valve leaflets at multiple time points. And we hypothesize that the anterior leaflets properties will change and the tissue will weaken and become softer over 5 hours. And now we're going to talk about our methodology.
Our first step once we receive the hearts is to dissect. And we must identify important anatomy and our alignment. So we look for the pulmonary trunk and we cut diagonally through there and we get the bottom view of the anterior leaflet seen here. And then once we get that bottom view, we can cut through the anterior and septal connections, and then we get a layout of the three leaflets. And then we have to extract the leaflet, but we have to be careful to maintain the papillary and annulus so that we can serve our orientation.
After we have extracted our leaflets, we can now do our biaxial mechanical testing, or BIAX for short. So first we measure the leaflet's thickness, and then we're going to trim, hook, and add markers to the leaflet. So in the top photo, you can see that the white chip is the phantom, and then we're going to add hooks all around it. After we mount our leaflet to the BIAX machine, we're going to run 10 cycles per protocol per leaflet for data collection. So we run 10 cycles, however, we only take the 10th cycle because the first few are kind of a warm-up for the leaflet to stretch. And we have 5 protocols that will stretch each axis in different percentage of load. However, in today's presentation, we'll only be presenting about equal biaxial. And after we collect our data, we can now do our analysis. Our first step is to obtain displacements from the markers using live tracking and digital image correlation. And then we are going to calculate and plot stress versus strain curves. And to do that, we're going to use 3 main equations that we input into MATLAB. So the first one is the deformation gradient tensor that uses the displacements from the markers. Next is the Green-Lagrange strain, which strain is the ratio of change of dimensions over initial dimension. And then we're going to use the second PK stress. Stress is also the ratio of applied force over area. And we can't use our standard equations that we might learn in school because the leaflet is a complex tissue and we need to account for the thickness of each sample and the length and be able to compare it to other studies.
To continue with analysis, our third step would be to use linear regression to estimate the upper tangent modulus, UTM, or Young's modulus, which is basically just the strain. And we use that region shown from 50 kPa to 120 kPa because it tends to be the most linear, so it's the easiest to create our tangent line. And our step 4 is to look at our statistical data by using one-way ANOVA tests to see if there's any significance with the data we collected.
And now we're going to show our results.
So these show 3 stress versus strain graphs at time 0, times 2, and time 5. So we did not include all 6 time periods. And our red is showing our circumferential, and our blue is showing our radial. And the shaded region is our standard error. We can see that the circumferential is a bit stiffer, and this is consistent with other studies. If you look closely, our N is decreasing over time, and this is because as time progressed, we happen to lose samples, whether that be because the sample didn't meet our standards in terms of protocols or because it ripped. And below is our average thickness.
So this graph next is modulus versus time, or UTM over time. And just as a reminder, the UTM is how stiff the leaflet is. And as you can see that the circumferential, or the bar graphs in orange, are greater or stiffer than the radial direction, which makes sense with our previous image and with previous studies. And using the one-way ANOVA test, there is no significant difference in UTM between time points. So that means that the mechanical properties do not change over time. Our next graph is the strains for physiological stress. So this is the percent strain at a specific stress level, or 85 kilopascals, and that's the amount of stress the leaflets experience when it's in systole or when the leaflet's closed. And this is kind of the opposite of the UTM. You can see the radial goes through much more strain and it's stretchier, which is why it's greater. And using t-test to compare, the mean radial and circumferential strains are significantly different at 85 kilopascals.
To conclude, once we've dissected the hearts and collected our data, we've discovered that there is no change in the mechanical properties of the anterior leaflet over our 5-hour time period. Now, we acknowledge that we had limitations within our experiment, such as our definition of fresh hearts, because obviously we did not butcher the specimen and use the heart straight from the butchery. The hearts were chilled, and then that's when we received them. And we had repeated protocols, and although the tissue should be able to withstand the repeated stretching, we acknowledge that there are times where we had to remount samples and potentially created micro tears. And we also only tested the anterior leaflet, which means we cannot conclude anything about the posterior and septal leaflets.
Nevertheless, we do have now a deeper understanding of the mechanical properties of the tricuspid valve, which leads to a new light and perspective on valve pathophysiology and biomechanics. Our next steps for this experiment are to do more sample testing, as you can see, and as Luca mentioned, we lost samples along the way. And also we have a few questions about like frozen hearts, if the mechanical properties change over time. We'd like to test other leaflets or other parts of the leaflet, and also their future applications to ex vivo experiments. And with that, we would like to thank everyone for listening and give a huge thank you to the Center of STEM Education and the YSP program, also to our lab and funding. This is our poster, and thank you again for listening.